Hey Kumar, can you hear me okay?
Yeah, I can hear you.
Perfect.
So we'll start by sharp 3 o'clock only, okay?
Yeah, perfect.
I think can we start now, Imran, right?
Yeah, sure. Sounds good.
Yeah. So, good afternoon, everyone. Welcome to our exciting live webinar titled Ensuring Drug Efficacy Solid Dose Testing Essentials, brought to you in collaboration with Scopely Scientific. My name is Dr. Kumar Jitendra, and I will be your host for today's session. We are thrilled to have Imran Hanif with us, a distinguished expert and business development manager at Scopely Scientific. With rich background in pharmaceutical quality by design, Imran specializes in solid dose, semi-solid, transdermal, and powder testing. His extensive experience in pharmaceutical quality assurance demonstrates his dedication to guiding customers through the industry's intricacies, offering innovative testing solutions for a wide range of pharmaceutical dosage forms. So before we dive into Imran presentations, I have few housekeeping things for you guys. Your audio. Please ensure your audio is muted during the session to avoid any background noise and ensure a smooth experience for everyone. We'll have a question and answer session at the end of the presentation. Please feel free to type your question in the chat box throughout the webinar, and we will address them during the QA session. This session is being recorded, and the recording will be made available to you after the session. If you encounter any technical issues during the session, please use the chat function to notify our technical support team. So without any further ado, let's welcome Imran Hanif as he talks us through the essential of solid dose testing and enhances our understanding of drug efficacy. Imran, over to you.
Perfect. So firstly, I want to say thank you to Kumar for the great introduction. I want to say thank you for everyone who's participating in today's webinar and for taking time out of your day. I really, really appreciate it. So yeah, I'm the business development manager at Copley Scientific. So I'm responsible for our solid dose testing as well as our powder testing equipment as well. And if not all questions get addressed, then never hesitate to reach out to me directly. So today I'm going to provide a quick guide to oral solid dose testing, and in a few slides' time, I will cover the agenda of what we'll be covering today. So I firstly thought I'll start off with a bit about us, a bit about Copley, for those of you who aren't aware of us. So Copley Scientific, we've been supplying equipment to the pharmaceutical industry since 1946. And we're global leaders when it comes to inhalation testing, as well as a trusted supplier for our customers' pharmaceutical testing needs as well. Our headquarters are based in Nottingham in the UK, and we have international partners and distributors around the globe. And India is a good example of that, in order for us to really help support our customers in the best way that we possibly can. And we are trusted providers of testing systems for pharmaceutical dosage forms, including tablets, which we'll cover in a lot more detail today, as well as capsules, transdermals, semi-solids, granules, and orally inhaled and nasal drug products. And we continue to work with industry groups and leading experts to bring relevant new products to the market. So in order to make sure that we're able to provide our customers with the best solution and an innovative solution for our customers as well. And that's backed by expert training and support for our customers. So today's agenda, so I'll be covering the gastrointestinal drug delivery, so just what it's composed of and the basics, the benefits of oral solid dose formulations, and the current regulatory landscape for oral solid dosage form testing. So I'll then go through an overview of the quality control tests performed. So I'll go into a detail about the apparatus, as well as how to conduct the test and the procedure that's required. And then we'll cover the advances in oral, then we'll cover the advances in oral drug delivery. So what's new on the horizon and what's popular within the industry at the moment. And this will be followed by, as Kumar mentioned, a live Q&A session where I'll be able to address some of your questions. So the gastrointestinal drug delivery. So the GI tract consists of the oral cavity, the pharynx, the esophagus, the stomach, the small and the large intestine, as well as the anal canal. And adequately assessing the drug product for any given delivery route is always required to understand the factors that will determine the efficacy and also the identification of appropriate in vitro test methods in order to get the most relevant data possible. And the GI route is a highly effective way of delivering many systemic drugs, and that's evidenced from the fact that 2/3 of all medicines prescribed today are oral solid dosage forms. However, designing oral formulations with predictable absorption is a major objective during drug development. So understanding, for example, the dissolution properties of oral formulations using like in vitro systems can really help support you on this endeavor. And we'll go into a lot more detail about that later on during this session. So firstly, the benefits of oral solid dose formulations. And solid dose formulation themselves, so the drugs, they play a crucial role in treating various diseases. So whether that be from cancers to autoimmune disorders and even genetic disorders as well. And the drug delivery via the GI tract is considered the preferred route of administration due to its convenience, its accuracy, its customizability, and the fact that it's patient compliance and non-invasiveness. I'm sure a lot of us on today's call would rather take a nice simple tablet than having to be injected by a needle. So, but there's also other ones that I've not included, which can be taken into account. So that could also be the physical and the chemical stability of solid dosage forms, which does favor it. And the oral drug delivery is a significant area of growth, and expected to be a growth of 10.5% from 2022 to 2030. Again, there is another however though, because delivering biopharmaceutical drugs orally does present unique challenges. And this is due to, for example, their large molecular size, their sensitivity to the GI environment, their poor aqueous solubility and membrane permeability, as well as inadequate absorption by the digestive system. But you can also take into account as well the chemical and enzymatic stability of drugs as well. So there are a few challenges which need to be overcome. So for oral dose formulations, we have immediate release. So this is where the drug is released straight away. Then we have sustained release. So maybe the drug will be released over a 2 to 3 hour period. We have delayed release. So maybe the drug won't take effect, for example, up until after 2 hours or so. And then we have biphasic and pulsatile release. So there are a few main goals in formulation design for the oral route of drug delivery. So firstly, you have local drug delivery. And this is, for example, to treat the GI disease where the drug is taken up into the GI mucosa, but it will not be systemically absorbed. And if it is absorbed, there'll be very, very minute absorption taking place. And systemic drug delivery, this is where the drug absorption needs to transverse the mucosal wall into systemic circulation. So drug absorption in the GI tract is governed by many factors, such as the surface area absorption, the blood flow to the site of absorption, the physical state of the drug, water solubility, and the concentration of the drug at that site of action. So we'll now cover the regulatory landscape for oral solid dose testing, and it's the responsibility, it's the responsibility for the safety, quality, and efficacy of medicines and medical devices does lie with various national regulatory bodies. So some examples would be the European Pharmacopoeia, the Japanese Pharmacopoeia, and even the United States Pharmacopoeia. And it's the pharmacopoeias that I've just mentioned that define the standards with which the drug formulation shall comply and the methods by which compliance will actually be adjudged. So moving on, we have the regulatory guidance. So the ICH is the International Conference of Harmonization, and they publish a single set of global specifications covering test procedures and acceptance criteria for new drug substances. And their purpose is to promote greater harmonization worldwide to ensure that safe, effective, and quality medicines are developed. But also the advantages, for example, if you have a manufacturing site that manufactures in Japan, it's the same company, so they have a site in Japan and they have a site in Europe. They may have different equipment because they're following different pharmacopoeial standards. So the way things are moving in that forward now is there'll be now greater harmonization. So there'll be alignment within those companies' SOPs, for example, and they can use very similar equipment and have the same procedures in place, which is a lot more convenient for the pharmaceutical industry. And harmonization is achieved through the development of ICH guidelines in 4 categories. So we have quality, safety, efficacy, and multidisciplinary topics. So over here on this slide, I've included a table which summarizes the different oral solid dosage form testing methods and which pharmacopoeia chapter they can be found in. If anyone would like a copy of this summary, this can be found in our oral solid dose testing brochure, and you can gain access to that online. So now I thought I'll cover the process analytical tools and the quality by design principle, something that's been quite heavily adopted by the pharmaceutical industry. So it's something that I thought that would definitely be worth mentioning today. So process analytical technology is defined by the FDA as a mechanism to design, analyze, and control pharmaceutical manufacturing processes through the measurement of critical process parameters that ultimately affect the critical quality attributes of an active pharmaceutical ingredient. But I know I've thrown a few random terms in there, but in a few moments, I'll go into them in a bit more detail. So quality by design ultimately allows for a systematic approach to the drug development process. And it's intended to improve quality, and it does this by using analytical and risk management methodologies for the design, development, and the manufacturing of new medications. And you can see I've included a little an image on the slide to just summarize the process. And it aims to basically design quality into the workflow from the outset, rather than just at the end of the process. And that can save pharmaceutical companies a lot of time, resources, and ultimately money. So the QBD roadmap is shown based on the ICH guidelines, and the basic elements include the identification of attributes concerning product safety and efficacy, which would be the QTPP, which is the quality target product profile, and the critical quality attributes. You can also use a risk assessment to link material attributes and process parameters to those CQAs, as well as the implementation of a control strategy for consistent process outcomes. And CQAs are defined as product attributes that are critical, like very critical for ensuring the safety, efficacy, and quality of the final product. And they must be carefully controlled and monitored throughout the tablet manufacturing process to ensure the final product meets its desired quality. So examples of this, which we'll cover in a lot more detail, will be the tablet hardness, its friability, as well as its dissolution and disintegration rate as well. So disintegration testing. So I thought this would be more applicable to start off with, and it makes sense because it's the first stage in the bioavailability cascade. So the regulatory landscape for the disintegration test. So before an API of an oral solid dosage form can be absorbed into the body, the tablet or capsule which they are contained must firstly disintegrate into smaller particles. And the tablet disintegration tester provides a repeatable in vitro test methodology and is an accurate way to carry this out successfully. And the test is carried out to determine whether tablets, capsules, or granules disintegrate within the prescribed time when placed in liquid medium under experimental conditions. And compliance with limits are stated within individual monographs, which can be found in the pharmacopoeias. So we now have the apparatus for disintegration. So the basket assembly has the basket you see on the slide, it has 6 open-ended tubes and 3 open-ended tubes in the case of larger dosage forms to accommodate larger tablets or larger capsules. And they're held vertically in position by 2 plates. You also need a suitable means to suspend the basket rack assembly from raising and lowering device using a point on its axis, a 1000 milliliter low form beaker, and the thermostatic arrangement for heating the fluid between 35 and 39 degrees. Also, the volume of fluid in the vessel It's such that the highest point on the upward stroke, the wire mesh at the bottom of the basket, remains at least 15 millimeters below the surface and descends no less than 25 millimeters from the bottom on the downward stroke. The system must also be capable of raising and lowering the basket in immersion fluid at a constant frequency rate between 29 and 32 cycles per minute. And you can also use a disc for each tube, if specified in the monograph, to aid disintegration to create that hammering action. But you would only ever use it if prescribed. So we now move on to the disintegration test procedure for plain and uncoated tablets. So firstly, you want to place the dosage unit in each of the 6 tubes of the basket rack assembly, and if prescribed, you want to add a disc. Next, you want to operate the apparatus using water or the specified medium as the immersion fluid, maintained at 37 degrees plus or minus 2 degrees. And the basket moves up and down in the vessel, which contains the 1,000 milliliters of simulated gastric fluid at 37 degrees through a distance of 55 millimeters. But it can be between 53 and 57, as you can see on the slide. At the end of the time limit specified in the monograph, you then want to lift the basket rack assembly from the fluid, and you want to observe all the tablets visually. And all of the tablets should have completely disintegrated. So in terms of the criteria, the pharmacopoeia does set a criteria to follow. So if all 6 tablets are tested and they all disintegrate, then you know your test has passed. If one of 2 of those 6 tablets fail to disintegrate, then it requires you to repeat the test on 12 additional tablets. So now you have a total of 18 tablets. So if 18 tablets are tested, the requirement is met if not fewer than 16 of the total 18 tablets are disintegrated. So anything passing more than 16, then you know your batch has passed. You can also apply this same test to the dosage forms highlighted in bold on the slide as well. But again, if anyone's got any questions in regards to other dosage forms for the disintegration test, then please don't ever hesitate to reach out to me. So now we have the DTGI series for disintegration testing, which we offer at Copley. And so I'll just go through the key benefits with you guys. So it's European, US, and JP compliant. So it meets pharmacopoeial needs. And it's really easy to maintain. So in terms of the water bath, it has the drain tap on there. And I'm sure a lot of us on today's call have probably seen a grim water bath in our time. So we have installed drain taps, so it allows for easy water drainage. We also have the easily removable water bath, which simply slides out. And we also have the easily removable disintegration baskets as well. So these can be taken to the sink and given a quick wash, ready to get ready for your next run. It has integrated precision temperature control measurement and adjustable stroke frequency controlled for accelerated of high sensitivity testing. And that makes it perfect, not just for the QC environment, but a great solution for the R&D environment as well. We also have 1 to 4 test station unit configurations, depending upon our customers' throughput needs. And the image you see in front of you is actually the DTG200IS. So it actually has independent station controls. As you can see, it has 2 independent platforms. And this can be really useful, especially when you want to assess formulations simultaneously. But for example, on the left basket, you could have an immediate release dosage form. And on the right basket, you could have a sustained release dosage form. So it allows pharmaceutical companies and other companies to test quite efficiently. And it also has single point electronic temperature calibration in order to make that whole process a whole lot less cumbersome for our end users. And I'll also say, again, it's very user-friendly, intuitive, cost-effective, and reliable for our customers. And we also have the heater, which is integrated not into the unit, but you can see that it sits nicely on top of the water bath. And if anyone's got any questions, please don't hesitate to reach out to me. So next, we have dissolution testing. And this is the next step in the bioavailability cascade, which we'll now cover, in order for it to enter systemic circulation and have the desired therapeutic effect. So the regulatory landscape for the dissolution test. So dissolution testing is the primary in vitro method for investigating and comparing the bioavailability associated with different dosage forms. And for absorption into the bloodstream, drug particles must firstly dissolve into molecular solution, and that's what is referred to as the term bioavailability. The measurements of dissolution rate really do support the optimization of bioavailability and consequently the therapeutic efficacy for a drug. And as you can see from the graph on the slide, it basically has a plot of concentration versus time, which is how you interpret the data. And it's considered a critical quality attribute for a drug product. And the dissolution testing technique can be used to predict the in vivo bioavailability or the assessment of bioequivalence for generics in R&D as well as QC, and can also be used with immediate, modified, or delayed release dosage forms, but also other dosage forms as well. So you have semi-solids, you have suppositories, and even topical and transdermal formulations as well. So in terms of the dissolution apparatus, so I'll mention in a second the different USP apparatuses there is, but the most common are USP methods 1 and 2. So they refer to the basket and the paddle. So the assembly consists of the following. So you have a vessel, you have a metallic drive shaft, which holds which holds the basket or paddle. You have the motor, the test apparatus itself, thermostatic control, and a water bath and heating device for immersing the vessel. And a fitted cover may be used to retard evaporation to prevent there from being any changes in fluctuation in the temperature. So make sure your results are accurate and reliable. So on this slide, I've included all the different dissolution test apparatuses used. So I mentioned the basket and paddle, so USP 1, apparatus 1 and 2. We also have the reciprocating cylinder and the flow-through cell, as well as the paddle over disc. And the paddle over disc is more used for your transdermal applications. Moving on, we also have this cylinder and a reciprocating holder. So the cylinder, again, would be used for transdermal applications as well. And the reciprocating holder is something that's been approved by the USP, but not found in the European pharmacopoeia. So as a total, there are 7 different apparatuses that can be used for the dissolution test. So in terms of the test apparatuses and methods, there's small volume conversion kits. So this could be really useful. For example, if you're working in R&D and you're working with something that's quite an expensive ingredient or an active ingredient, for example, then you can use 100 ml or 200 ml volume vessels. And even if you're in QC, for example, and you're working with something that's quite potent in nature, again, having a small volume conversion kit can be extremely useful. And the dimensions of the vessel and paddle are in proportion, and it consists of the miniature vessel, the miniature paddle, a miniature vessel cover, and a special centering ring assembly. So everything you need to carry out a successful run using smaller volumes. So now I'll move on and cover the dissolution test procedure for immediate and extended release tablets. So firstly, you want to place a stated volume of dissolution medium in the vessel of the apparatus, which is always provided in the monograph found in the pharmacopoeia. You then want to equilibrate the dissolution medium to 37 degrees plus or minus 0.5, and you want to use the medium specified within the monograph. So step 2, you then want to place 1 dosage unit in the apparatus, taking care to exclude any air bubbles from the surface, and immediately operate the apparatus at a given specified rate given in the monograph. Step 3, within the time interval specified, you want to withdraw a specimen from the zone midway between the surface of the dissolution medium and the top of the rotating basket or paddle, ensuring it's not less than 1 centimeter from the vessel wall. And where you need multiple sampling, you want to replace those aliquots with fresh dissolution medium at 37 degrees, unless it mentions not to carry this out. Then lastly, you want to perform the analysis directed in the monograph using a suitable assay method. So whether this be UV absorption or whether this be HPLC. So you can see dissolution is a great way in order to prepare that samples that's required, in order for analysis using that suitable assay method. So on here, on this slide, I've included a table which shows all the different USP apparatuses for this solution and which dosage forms they apply to, as well as some key process parameters. And I've just noticed, actually, I didn't go through and mention what critical process parameters are, but they're parameters or manufacturing steps which really need to be kept, I would say, kept quite refined in terms of you want to make sure that there's a tight threshold in order to meet the desired quality and the best quality possible for that final product. And if anyone's got any questions or if anyone would like a copy, then please reach out to me. I'd be more than happy to send this to anyone if they request it. So now we have the DISI series for dissolution testing, and it's used to assess bioequivalence for generic products and for the confirmation of batch-to-batch equivalence in QC. We have two different models available. So the model you see in front of you, the 600i, where you have the 6 vessels with the integrated heater. The 800i is the integrated heater is replaced by the 2 additional vessels, and you have an independent heater instead. But again, it's still got quite a small it still offers quite a small footprint. It's European, US, and JP compliant, and you have accessories to provide a wide range of solutions. So you've got the small volume conversion kits, for example, you've got the automatic tablet drop, and you've got various methods of sampling as well. You have integrated precision temperature control measurement and a wide speed range to accommodate a broad scope of methods. Again, it's just something that makes it perfect for R&D and not just for QC applications. As I mentioned in the beginning, it does have 6 to 8 test station unit configurations and single point electronic temperature calibration control as well. But like the disintegration tester, it's very user-friendly, intuitive, and we've had great feedback from the industry so far. So it's a great solution for those QC testing needs. So moving on to friability testing, which is a physical test for oral solid dosage forms, which we will now cover in more detail. So friability testing is a tendency for a tablet to chip, crumble, or break, and is one of the critical quality attributes used to quantify the physical integrity and stability of an oral solid dosage form. It is typically caused by poor tablet design, low moisture content, and even insufficient amount of binder. So optimal friability is really essential because you want to make sure it has sufficient physical stability for transport and storage, while at the same time, you want to allow it to break down readily in the GI tract for the subsequent processes, so for your disintegration and dissolution. Oh, sorry about that, guys. And a weak tablet could be due to a number of factors, including poor tablet design, low moisture content, as I mentioned, but even due to over-lubrication, which can lead to breaking as well as cracking. So in terms of the friability apparatus, you want to use a drum made of transparent synthetic polymer with polished internal surfaces. One side of the drum must be removable, and the tablets are tumbled at each turn by a curved projection, and it has an inside radius between 75.5 and 85.5 millimeters. The outer diameter of the central ring is between 34.5 and 25.5 millimeters, and the drum is attached to its horizontal axis and rotates from 24 to 25 RPM. So in terms of the friability test procedure for compressed and uncoated tablets, which we'll cover. So for tablets with a unit weight of more than 650 milligram, you want to take a sample of 10 whole tablets and dedust. In the case where it's less than 650 milligrams, you want to take a sample of close to 6.5 grams as possible. You then want to accurately weigh the tablet sample and place it in the drum and rotate the drum 100 times using a speed between 24 to 26 RPM, and 25 is the most commonly used. If the tablet size or shape causes irregular tumbling, you want to adjust the drum so it forms about a 10-degree angle with a horizontal. And in a few slides' time, I'll show you how that's possible. So in terms of the criteria, if the tablet is cracked, tablets have cracked, cleaved during the test, then it's failed. If it's difficult to interpret, then you want to repeat the test twice and take a mean of those 3 tests. And the weight loss from a single test, or the mean of those 3 tests carried out, it needs to be less than 1% in order to pass. Anything above 1%, then you know your batch has failed. So we offer the FRVI series, which is a unique solution for your tablet friability testing. So we have 2 different models available. So the model you see in front of you is the 200i with 2 drums. So on the left-hand side, you can have a standard friability drum. And on the right-hand side, you can have an abrasion drum to create like a harder drop for those tablets if required. But if you wanted to, if you had quite a bit of throughput to achieve, then you can use 2 drums if you wanted to as well. It's EU, USP, and JP compliant. It has convenient drum emptying functions as well as drum loading functions as well. So when you're ready to load your sample, you'll click the button on the far left, and it will bring the drum. It will rotate the drum round to you, and you insert your tablets. And it also has a drum emptying function. So you press the button, and it will automatically drop the tablets for you into the tablet tray, which sits below the drum. So it doesn't require too much human involvement, and it makes it easier for our end users as possible. It also has integrated calculation of percentage weight loss, so it reduces the chances of any human error, and it's just quite a nice feature just to make end users' life a lot easier. And on the bottom right, you'll also see the built-in 10-degree angling device for larger tablets in the case of irregular tumbling. So you just simply kick up the kickstand in order to use that for those applications. And it has, again, like our other product, all the products in our range, it has an intuitive touchscreen, and it simplifies the operation, and it clearly displays all test parameters throughout the run for the end users. So now we have tablet hardness testing. And this is another physical test and a critical quality attribute of a tablet used alongside friability testing. So the regulatory landscape for the tablet hardness test, it allows for the quantification and the hardness/robustness of the tablet. And it's more correctly defined as braking force in the USP or crushing strength in the European Pharmacopoeia. And it's used to quantify the physical stability of an oral solid dosage form. The test is intended to determine under defined conditions the resistance to crushing tablets, measured by the force needed to disrupt them by the crushing or the braking force required in order to produce failure. And hardness is a CQA that tablets really must withstand vigorous handling and transportation experiences in the manufacturing plant, as well as in the drug distribution system and the field of end users. At the end of the day, you want to make sure you ensure accurate dosing for those patients. And the tablet strength serves as an important criterion by which to guide product development as a QC specification. And high hardness values indicate increased disintegration, as well as reduced friability, as well as reduced dissolution and increased friability values. So in terms of the apparatus, it consists of 2 jaws which face each other, one of which move towards the other. One of the jaws is attached to a force gauge, and the other is attached to a motor, providing the mechanical drive. And then the motorized jaw drives forward, pressing the tablet against the fixed jaw until the tablet breaks. In terms of the hardness test procedure, it is quite simple. The tests to carry out. So step 1, you want to place the tablet between the jaws and orientate the tablet in the same way. One of the platons will move towards to apply specific force to the tablet, which will then cause fracture. And you then want to carry out the measurement on 10 tablets, taking care to exclude all fragments and make sure they've been removed. So you just have to dedust before each determination. And you want to express the results as the mean, minimum, and maximum values of the forces measured. But there are also quite a few considerations which need to be taken into account when carrying out this test. So, such as the rate of uniformity of loading, using a constant force, as well as the tablet orientation and sample size. So we offer the TBF100i for hardness testing, which is our really innovative solution for our customers. So it's EU USP compliant. You have a choice of braking force measurement units. So whether that be kilopons, pounds, or newtons. You have automated entry of tablet weight and thickness. So you can refer to our website, but you can also get a thickness gauge, which has been customized to sit on top. So let's say you place your tablet in the thickness gauge. It will measure the thickness. You then click the play button on the hardness tester, and that value will automatically be inputted from the thickness gauge into the hardness tester. And the same also applies for analytical balance as well. So it's a great integrated solution for our customers. As you can see, parameters are very clearly displayed, and you can also print from a click of a button as well. So on the right, you do have the thermal printer, which you just stick into your lab book, and it can do batches up to 99 tablets per batch, and it provides you all the data that's acquired as well. And we provide everything that you need in order to have a successful run, as well as accommodating a wide range of tablets. So we also have the TH3 series for hardness testing. So, and it's ideal. So essentially, you'll place the tablet in between the load cell, and then you'll use the knob at the bottom, and you'll turn the knob using your hand manually, and it will then push against the tablet until it causes fracture. And the maximum value will then be displayed on the LCD screen. So it's easy to calibrate. You have a choice of units. It's portable with a clear LCD display. And it can be really useful, especially if you have a large QC lab. You have a QC lab, for example, it may be upstairs, another QC lab downstairs, or even in different rooms on the same floor. It's just really convenient in terms of carrying it with you and taking it to wherever it's required. So it's quite a popular option for our customers. So now we also have the advances in oral drug delivery, which I'll now cover. So what's on the horizon and what's been quite popular. So oral administration is the most convenient route of administration for drug delivery because it offers high patient compliance for the reason we mentioned during the beginning of this webinar. The drug pipeline is making formulation as a simple tablet unviable due to some of the challenges that we mentioned. And the pharmaceutical industry screening programs identify that around 40% of new chemical entities face various difficulties at the formulation and development stages. So we have amorphous solid dispersions. So drug candidates use amorphous solid dispersions in their formulation to help overcome the solubility limitations and enhance the rate of dissolution. And the system comprises of an amorphous active pharmaceutical ingredient, stabilized by a polymer matrix to provide enhanced stability. And the amorphous solid dispersion maintains its supersaturation in the gastrointestinal tract, improving the bioavailability of the drug. So we also have oral biologics. So biologics from monoclonal antibodies and peptides to gene therapies. They are delivered by injection or infusion because of their susceptibility and degradation within the GI tract itself. So advances in oral solid dosage testing means that some biologics, such as insulin, can now be developed for oral drug delivery. An example of this, which has been approved, is Novo Nordisk Rybelsus, an oral formulation of semaglutide for treating type 2 diabetes. And it represents advancements in oral drug delivery. And I definitely see this as a great way of where the market is moving, and we can only expect growth within this industry. And we also have dissolution, which is the most sought after drug release technology, and it's one of the most popular drug release technology systems on the market. And it's never had a decrease in need. There's always been an increase in need for the system. And this could be due to the rise of generic formulations, and this makes it remain dominant within the industry. I know I'm moving slightly a bit off topic, but it's not only just being applied for oral solid dosage forms, but there's been recent advancements and improvements in making it approved for nasally inhaled drug products, for example. So there's a large and a wide scope for dissolution, and I can see it always being there's always going to be a need for dissolution testing. So it's quite exciting to see what else happens in the future as well. So now just a bit about Copley now in terms of our training services. So we offer a great variety of training courses for our customers. We have experienced trainers, as well as Claire, who's our application specialist. And we have a modern and fully purpose-built facility. So please feel free, if anyone ever wants to come down to on-site, please don't ever hesitate to reach out to me. And it'd be great to meet you in person. So I just want to finally just say a really big thank you for everyone for participating in today's webinar. I'll now have a great opportunity to address some of your questions, which I really look forward to. But again, just a really big thank you, and thank you for taking time out of your day. And I'll hand back over to Kumar.
Hello, everyone. Thank you all for being here. So now it's like time for Q&A session. So dear all, if you have any questions or clarification regarding whole presentation presented by Mr. Imran, so you can type your question here on Q&A chat box. So I think we have one question, Imran sir.
Yeah.
The question is by Mr. Suresh Babu.
Yeah.
So yeah, he's asking why cannot we go for above 50 RPM for type 1 apparatus?
Is that for dissolution?
Sorry?
Sorry, could you repeat that again? Sorry.
Yeah, he's asking why cannot we go for above 50 RPM for type 1 apparatus?
Oh, type 1. So if that's in regards to dissolution, it's just each drug product has its own monograph. So it has its own set of specifications to follow, basically. A lot of what I've provided today is like a general overview and the most common use settings. But again, if you look in the pharmacopoeia and it states that you need to use something else, then you just follow the pharmacopoeia, to be honest with you. So that's the best way to do that. So it literally just depends. And if you're in an R&D environment, you can pretty much use whatever speeds that you would like to use. It's just with a QC, you just have to make sure that you're complying with the pharmacopoeia.
Okay, thank you so much. Thanks for the answer. Hope you're satisfied with the answer, Suresh sir. Still, if you have any questions or clarification, I have mentioned one email ID here in chat box, Q&A box only, which is rajiv@apexchrome.com, or you can directly write to me or like Imran sir also after the session. So here one more question, sir. I deal with early stage drugs when we only have access to small quantities. What option do you have for dissolution?
Of course, yes. Yeah, so it's something that I did cover as well. So basically, we offer the 100 ml and the 200 ml conversion kits. And as I mentioned, the way this could be really useful, so it's useful for QC in terms of, especially if you're working with a drug that's quite potent in nature, but as well, it's great for the R&D as well. So if you're working with something that's quite expensive and you don't want to use too much of it, then I would always recommend using the 100 or 200 ml small volume conversion kits. So we'll always have a solution for you. And if anyone's ever interested, please don't ever hesitate to reach out to me. And if anyone wants to discuss the application in more detail and get some advice, again, don't ever hesitate to reach out, and I'll be more than happy to organize a Teams call with anyone who's interested.
Okay, so here another one, sir. We often assess multiple formulation, such as immediate and sustained release. What's the best way to generate comparative disintegration data?
Perfect. Great question. So I think the best way to do this would be the best solution that we offer, and quite an innovative solution on the market, is the DTG200 IS. So as I mentioned, you can use both baskets. So one basket could be used for your immediate release, and the other basket can be used for your other dosage form. So your other release mechanism. So whether that be delayed release or even sustained release. And then what you can do is you can observe the tablets and see when they disintegrate. And then you can also just plot a graph, and you can basically compare the data. So compare those two different formulations and gain some sort of conclusion from that.
Okay, sir. Thanks for the answer. Here is one more. If you work with granules and pellets as well as solid dose, how could I conduct friability?
Oh, perfect. No, brilliant question. So it's something I actually didn't cover in today's presentation, but it's something I'll be definitely more than happy to cover with you now. So you would be for harder dosage forms, you're going to be unable to use a conventional friability tester because it's not going to make a considerable change in the surface mass. So basically, you need a higher energy input instrument, which is actually needed to generate that quantifiable change in surface mass. So a solution that we offer is the Frabima SA4000. So please refer to our website and our brochure, and you'll be able to find this on there. But essentially, it's like an enclosed system, which has an oscillating arm where you insert your container. So essentially, just to go through the procedure with you quickly, is firstly, you just want to remove any fine particles using a sieve. In a glass container, you want to have about 10 grams of your granules or spheroids, and you want to install the container in the apparatus, so locking it into the oscillating arm. And you want to shake for 240 seconds at the highest frequency for harder granules or spheroids. Or alternatively, you could do it for 120 seconds at a lower frequency for softer granules or spheroids. You then want to sieve and weigh the granules, and then you want to test 3 samples and calculate the mean value. So that's how you'll carry out the test. And you just no worries at all. Thank you. Appreciate the question.
Sir, one more, can I get a printout with the statics directly from the TBF 100i?
Oh, yes. So the image I included on my slide actually wasn't the best image, but on the right-hand side of the TBF, there's basically a thermal printer. So from the click of a button, you'll press the print report button on the TBF 100i, and automatically you will get a printout, and you can just tear it off like a receipt, like you get in the shops, and then you stick it inside your lab book. So everything's integrated into the equipment for you. And that can be really useful instead of having a printer which takes up a lot of space alongside, because we all know nowadays lab space has now become a premium. So having the printer integrated, we've only ever had great feedback from our customers, and it's something that they really enjoy. Yeah, and it's a solution that for your printing needs that will suit your needs quite well.
Okay, sir. Thank you. One more here, sir. Does the FRV range automatically calculate the friability value?
Yes, sorry if I didn't cover this in a bit. I make this clear during my presentation. However, in terms of the friability, so you will literally click on the calculator button, which is basically like on the home screen, and then you would insert the tablet weight prior to friability testing. So let's say, for example, you had 10 grams of tablets, then you rotate in the drum, and let's say you have 9.99 grams of tablet now. So you then weigh the tablet, after you've spun it in the drum, and now let's say you have 9.99 grams. You'll then insert that onto the calculation screen, and it would automatically calculate that friability value for you. So if it's less than 1% less, the tablet has passed. But anything above 1%, your batch has failed, and you need to look and do some more development work and look into your formulation a bit more closely.
Yes, sir. Dear all, if you all have still any questions, please type it into Q&A box.
And just to add, if anyone's got any questions, please feel free to reach out to me on LinkedIn as well. I'll be more than happy to address any questions that anyone has.
Yes. You'll also get like feedback mail after the session. Still, if you have any clarification, you can reply to that mail, and we'll forward your questions to Stream of Copley Scientific and Apex Chromatography, and you will get the reply along with detailed answers and clarifications.
Amazing. I look forward to receiving those. Appreciate it. Thank you.
Yes, yes. Thank you, Suresh sir, for asking question. We'll wait for another 4 or 5 minutes. Still, if you have any clarification, please type it.
No worries.
I think there is no any, sir. So
No, that's fine. If anyone's got any question, they can reach out to me directly, or
yeah,
reach out to
you
yourself,
can,
and you can send them
yeah, they
across to me.
can. You will get like a thank you mail after the session. You can reply your question there also. Mark CT2 like. So I think so. Say thanks. Thank you everyone for joining us today. Thank you, Imran sir, for giving your precious time for this session, insightful session.
It's been a great
Yeah,
opportunity to get insight and talk to the Indian market. So I appreciate
yeah.
it.
Stay in touch with like Microbios India for like many more such insightful session in coming weeks and months.
Amazing.
Yeah. So thank you again, Imran sir.